Hey guys, it's Miss Hendricks. This is still season 0. This will be episode 2 for IA, and this will be episode 1, I believe, for AP. And this is about experimental design and metrics. This is important for all students to understand in biology because you have to run both a fair and a valid experiment so that others in our field can run the same experiment, get the same results, peer review, all of those important things that we're going to learn about throughout the year. So 4 things we're going to cover, 5 things for AP. 1, we'll talk about experimental design terms that you need to understand. 2, we'll do an example. 3, we will cover basic metrics. 4, we're going to go through a metrics example. And then 5, for AP, we're going to apply this to our fruit fly behavior lab. So for terms, first we have independent and dependent variable. Dependent is the data. It's what you are measuring, right? If you remember back for Intro to Advanced, that would be the y-axis. It is our data. It is what you're measuring. You can remember it. It's the DRY, right? Dependent responding y-axis. Independent variable is what I am changing. It's what you're testing. It's what you're manipulating. It's what you're altering to figure out if it affects your data that you're going to be getting. So independent was the x-axis. You can remember that MIX, right? Mix. Manipulated independent x-axis. Then you have your control group. That is what you're going to compare your results to. So it does not have the independent variable. So if you were trying to test the effects of fertilizer on plant growth, you would have one plant without any fertilizer to see how it grows normally, and then you would have one plant with fertilizer. You might have multiple groups with different levels of fertilizer to see how those different levels affect plant growth, but you need to have something to compare it to. And then lastly, you have constants in your experiment, and these are also called control variables, not a control group. Easily confused. That's why I often call them constants. And it's what you keep the same. You need to be really specific when talking about these. Don't just say same light, same temperature. Talk about specifics. The room temperature, right? Talk about what type of light. Is it natural light? Is it artificial light, like in our classroom? Give me specifics on those. Same amount of water. How much water are you giving them? But those are things you keep the same so that you know that they're not affecting your results. For an example, a question could be, are Bounty paper towels the strongest? So the dependent, what we're measuring is the strength of those paper towels. And the independent, or the thing that we're changing, is the type of paper towels. So specifically, we are caring about the Bounty paper towels, right? And we are going to have a control group of other types of paper towels so we can compare the Bounty paper towels to that control group. You could also use Adidas shoes if you were trying to figure out if Adidas shoes make you run faster. Think about what the dependent variable would be for that one. What are you measuring? Well, obviously, if you want to be running faster, you'd be measuring the speed. And that means that the independent variable would be specifically Adidas shoes, right? You would think the type of shoe, but independent, you're trying to figure out if Adidas shoes are faster than the control other types of shoes, right? Now going into the metric system, I'm sure that you've all heard of King Henry died by drinking contaminated milk. And that's why I have kilo, hecta, deca, base, deci, centi, milli. Base is just that standard middle, right? So base, I put on here, can include any one letter system of measurement that would be a base unit. So that would be grams, which is G. It could be meters, which is lowercase m, or it could be liters, which is capital L. And the reason I have 2 spaces to the right of Millie is there's 2 spaces in between Millie and this weird U I have. It's actually a mu. It's a weird letter M, but it doesn't have a symbol in the computer, so we often represent it as a U, and that's micro. So the way that the metric system works is it moves by units of 10, which is lovely, because then that usually means you can just move the decimal. So that's why there's those 2 spaces in there, so that we can remember to account for those 2 spaces. So I have some examples that you can work through, and there's going to be some questions that you need to answer about them. But if I was given 476.79 centimeters, I would go to the centimeters, and it wants me to go to meters, right? You see how I see the letter M? Don't confuse that with milli. Milli would be shown with 2 letters, millimeters, MM. So what it really wants me to go to is our base unit. So the way I would do that is I'm going to be moving my decimal place 1, 2 units, to the left. So that would give me 4.7679 meters. Okay. I want you to try the other one by yourself. It's converting 0.0067 liters to microliters. So remember, include those spaces. And liters, you should remember, is a base unit. So start with the base and move all the way to micro. Hopefully you figure this one out by moving it 1, 2, 3, 4, 5, 6 to the right. So you should have got 6,700 microliters. If you need any additional practice on that, there's lots of resources online, or I can help you out with that. So, intro to advanced students, you should be able to identify different variables in an experiment, including independent, dependent, control versus experimental groups, as well as constants or control variables is another word for it. You should be able to design your own experiment using these variables, and you should be able to convert using the metric system. Remember, all life is an experiment, and the more experiments you make, the better, which was by Ralph Waldo Emerson. If you are one of my AP students, we have some additional information to go through that applies specifically to your fruit fly behavior lab. So the first thing I want to talk about is what we're trying to really research, or what information we're trying to get out of the fruit fly behavior lab. And that is basically fruit flies preference, right? You are designing your own experiment. So I'm just going to give a more vague example. That's why I said, what do fruit flies prefer? But I'd want you to give specifics on your research question. Do they prefer this over this? And then I need you to make a prediction about what you think they will prefer. I think they will prefer, well, don't use I, but fruit flies will prefer blank over blank. And then you need to write a hypothesis based on that prediction. If fruit flies are given vanilla syrup, then fruit flies will be attracted to it because it's attractive to their olfactory senses, etc., whatever you find in your research, right? I like if then because statements, because if independent variable, so if I change this variable, whatever I'm trying to measure, whatever I'm changing between my experiments, then my dependent variable will happen, whatever I'm measuring will happen because scientific reasoning behind it. What I've included on here is a picture of an empty straw. And if I was trying to test if fruit flies prefer that vanilla syrup I mentioned in my hypothesis versus just sugar water, I would first need to test a couple of things. I would need to establish controls in the sense of positive and negative controls. So negative controls are something that you expect no results out of. So for my negative control in the fruit fly experiment, what would be something that I would not expect a result to happen? I would not expect preference. I would not expect anything to happen. That would be if the tube was completely empty, right? If fruit flies randomly started generating into my tube, that would be an issue, right? So negative control would just be running the experiment with an empty chamber, right? And that just allows you to test to make sure your equipment isn't faulty and your results are valid, right? Your test that you're running is valid. It's not randomly generating flies when it shouldn't. Then for the positive control is something that you would expect a positive result on, right? So your positive control or your positive control would be having no independent variable on both sides. So that'd be having sugar water on both sides. You would expect a result, right? You'd expect a positive result. They would prefer both of them equally. This is different than your control group, because your control group is the sugar water, right? That sugar water allows you to compare to the experimental group. The experimental group would be your vanilla syrup, right? So your control group would need to have the same concentration of sugar as the vanilla syrup so that you know that the sugar as well as the water is not the reason they prefer the syrup. It would be the vanilla part of it, right? So you have vanilla syrup on one side, you have sugary water on the other side with an equal amount of sugar and water as the syrup, so then you know they prefer the vanilla that you're testing. Then you would have the constants, the things you keep the same in the experiment. Just like I told Intro to Advanced, you need to make sure that you are specific, right? Same conditions, same lighting, same temperature. Give me specifics. What is that temperature? What is that lighting, etc. The dependent variable that you're going to be measuring is, do they prefer the sugary solution or do they prefer the vanilla syrup? So you're going to be counting the number of flies on either side of the chamber at a given amount of time. Say you set it for 3 minutes. At 3 minutes, you're going to measure and see how many are in each side of the chamber to see their preference. You are going to be designing your own fruit fly experiment to figure out what their preference is between two different things. Make sure that you keep in mind what you're going to be keeping constant so that your results are not being affected by anything other than the independent variable you are trying to test. Make sure that you understand positive and negative controls. And if you have any additional questions about your fruit fly experiment, please let me know. Don't forget to take chances, make mistakes, and get messy. Thank you so much.